Uh, okay. It's a pleasure to introduce our second seminar of this semester, and just a small advice, a small announcement. From now on, all the seminars will be available in this YouTube account, Comunicação NeuroMat, and you can find it in the same link that we are making available in our Facebook.
In our Facebook page, I believe.
In our Facebook page. And all the seminars will be available in the same link. So, and you can find it in the YouTube channel also. The seminar of today is our new postdoc researcher, and he will speak about implicit short-term memory and contextual biases. And his name is Raimundo Machado de Azevedo Neto. This is it. Welcome to our team. And
Thank you, Aline. So for those of you who don't know me, I'm Raimundo. I've done my PhD at the medical school here at the University of São Paulo at the radiology department. So my background is basically in experimental biology, or actually in cognitive neuroscience, but doing experiments. And today I'm going to show you part of the work that I've done during my PhD, some part here at University of São Paulo and some part at Universität Tübingen and at the Max Planck Institute in the city of Tübingen. And in the second part of my seminar, I'm going to present some of the work I've been doing after my PhD and what I intend to do here as a new member of NeuroMat. So the first part of my seminar is going to be about visuomotor integration, which is basically the first part of my PhD, and then short-term memory, which is, I think, the longest part of my seminar, which will lead to a class of models that I've been trying to learn and use to explain my experimental data, and then try to make a link between what I've done using, trying to understand short-term memory and what you've been trying to do, how we learn from statistical regularities in our environment. So what is visuomotor integration? So we live in a world that we interact with. And most of our interactions are through the visual senses. So in order to act upon a cup of coffee, we have to perceive where this cup of coffee is in our environment. And we don't have just to perceive the object and know that it's coffee to interact with it. We have to estimate where the object is so we can grab the handle of the cup of coffee and bring it to our mouth. And we do it effortlessly in our daily lives, but this is a very complex processing that our brain does. And we still don't know exactly how it's done, but it's a very complex computation to be performed. So as an example, we don't have robots that can do it neatly as we do. So 2- or 3-year-old child can do this very fast and very easily, but a robot cannot do it. And to make things worse, we don't live in a world where there is just stable and static cups of coffee, but we live in a world that is always changing and moving. So when we cross the streets or drive or play tennis or baseball, we are dealing with moving objects. And we don't have just to estimate the position of the object, but we have to know when this object will be in a certain place in order to interact with it. So one way that has been used to simplify these kind of dynamic tasks in dynamic environments is called interceptive actions. It's a simple class of actions in which we have to estimate when and where a simple object is moving and try to grab it or try to hit it or try to avoid it. So it's a rapid motor action toward this moving object. And to make things even more complicated, when we process visual information, this visual information takes a couple of milliseconds to be processed in the brain. So from stimulating the retina, going all the way to the visual cortex, to the hierarchical stream of information in the ventral visual areas, And to generate a motor command, it takes around 200 milliseconds. If we would judge our environment based on this information, we would always be late with respect to the objects. For instance, if you're playing tennis and someone hits you with a serve at 160 kilometers per hour, you're going to miss the ball by a couple of meters. And this is not what happened. What we experienced, that people is actually able to act upon objects that were there in the past. So we actually predict the future, so we can act upon the objects. So to overcome this visual motor delay, we have to conceive that the brain uses some kind of anticipatory mechanism, a predictive mechanism, that tries to understand when an object and where the object will be in the future. And although parts of this puzzle have been investigated, like how the movement is processed in the brain or how we control movement, are already being discovered by scientists, we don't know much about how these two informations are gathered or integrated in order to predict the motion of objects. So this is an example of an experiment where we try to understand how we perceive and predict where the object is going to be. So participants have to look at a ball that is traveling toward an object, and this object is decelerating toward the wall that is right here. And then suddenly the object, it just disappears, and the subject has to judge whether the object would hit the wall if it had continued its path toward the wall. And in this kind of task, when you have to predict the time that the object is going to hit the wall, what we usually see using functional magnetic resonance imaging is activation in specific areas of the ventral premotor cortex and the inferior parietal lobe, here specifically the supramarginal gyrus. So this has been consistently shown in a couple of experiments that when we try to predict time using a perceptual task, we have activation in the ventral premotor cortex and in the supramarginal gyrus in the left hemisphere of the brain. So this suggests that in order to predict objects, we need these areas to be active. But one difference that is from these protocols, from our daily experience with objects, is that in these scenarios, participants, they have to perceive the object and just judge whether it was going to hit the wall or not. And usually when we are crossing the street, we are trying to avoid an object to hit us, or when we are playing tennis, we are integrating the motion with our own motion. So we ask whether the regions associated with this integration between motion and our movement are which brain regions are associated with the integration of those two components, motion of the objects and our own motion. So to test this, we design an event-related functional MRI experiment. For those who are not familiar with fMRI, we use a proxy for neural activation, which is based on the blood flow, blood volume, and the concentration of oxygen in the hemoglobin. So these images are acquired throughout the time, while participants perform a couple of tasks. The main task we have is an interceptive task. So participants, they see a dot moving from one side of the screen to the other, and they have to press a button at the same time as the target hits the wall, as if the participant was catching a ball. So if the ball comes to your hand, you have to close your fingers at the same time it hits your palm. Otherwise, if you close it too quickly, you are too early. If you close too late, you lose the ball because it hits the palm of your hand and it goes over your hand. So this task simulates this kind of behavior. And when doing fMRI, we have to control for activation related to other aspects of the task. And this is what we have done here. In a control task where participants see the same stimulus, but they only observe the motion of the stimulus. They only pay attention to the motion of the stimulus. And in another control task, we control for the motion of the participant. So they only have to react to the change of the dot they are fixating throughout the task. So they have to fixate this dot all throughout the experiment, and they have to press the button once this dot transforms into a ring. So we then control for more simple processing for motor and visual areas. And to analyze this data, we analyze a sort of multiple regression on every voxel of the brain, which are the volumetric pixels we get from fMRI, so throughout the whole brain. So our hypotheses with respect to this task were based on previous studies on perception. So if perceiving the motion and judging whether it's going to hit the wall and acting upon it use the same mechanism, we would expect to see brain regions activated in the left hemisphere, more specifically in the left supramarginal gyrus, here in the inferior parietal cortex, and in the ventral premotor cortex. And also we would expect to see activation in the interparietal sulcus, here between both lobes of the parietal cortex, which are associated with integrating visual information of static objects. So we would expect to also see this integration for moving objects, not only static objects. And what we get from this experiment is, instead of lateralized to the left hemisphere activation, we get activation on both hemispheres of the brain. So here we can see activation on both hemispheres, so not only on the left side. And instead of the ventral premotor cortex, we get activation on the dorsal premotor cortex. We get, yes, activation on both intraparietal sulcus. Here you can see it, here you can see it. On the back of the brain, on the posterior parietal cortex. And at area V5, which is part of the visual cortex, an area specifically associated with visual information of motion. So from this experiment, we got that instead of lateralized frontoparietal network, but ventral, we found a bilateral dorsal frontoparietal network, including the human V5 complex, which processes visual information of motion, and the angular gyrus in the parietal cortex. And these areas are more associated with integration of motion than simple processing of visual motion or motor processing. So after investigating these regions that are associated with integration of visual information of motion and action, we try to understand how we deal with different contexts. So in our daily lives, we are not doing our actions completely isolated. We are doing our actions in an ever-changing environment, and we have to take into account these changes. And a very nice and very replicated, many times replicated effect is known as previous trial effect, but some people also call it serial dependence or decision inertia. There are many names there by which this phenomenon is called, which is the effect that the immediately previous movement or perception that you had influence your perception or your action in the next trial or in the future. So it's the effect of the preceding trial on the next trial. And this is very neatly presented here on a simple experiment where participants see a ball moving along a path, like in the experiment I just showed you, and then this ball, it disappears behind an occluder. So behind this occluder, it never appears back, and participants have to judge when the ball would leave the occluder and appear on the other side. So they have to estimate the time it would take to travel behind the occluder and be right about this time. And what we get when we analyze the temporal error, so when they hit too early, before the target would appear after the occluder, they have negative errors. They are too early. And when they press a button after too much time after the target left the occluder, they are late, and then their error is positive. And a very nice thing is that given the previous trial, their error is different. So if you wouldn't expect any effect of the previous trial, their error should be all around zero or all around the same bias, either positive or negative. But what we see here is this slope showing that if the previous trial was like 2 degrees per second with respect to a reference trial, which is a target traveling at 20 degrees per second. If the previous trial is too slow compared to the current one, participants tend to be late. If the previous trial was faster than the current one, participants tend to be early. So this bias is very neatly shown here from one trial to the next. And one proposal for how this previous trial affects the current trial is that the previous velocity, the previous speed of the target is joined with the previous one is joined with the current one. So both speeds have some weight in this weighted average, and then the current estimate of the speed is based on this weighted average of previous and current speed. But for the previous speed to have some effect on the current one, you have to store it somewhere. You have to keep it from one trial to the next. And in order to occur this, we have to conceive that the brain is using short-term memory to integrate both current information and previous information. And then if the brain stores this information, we would like to know which regions of the brain are storing this information we are dealing with in the environment. So which regions of the brain store this information in short-term memory? According to a recent proposal, actually a couple of years now, like 5 or 6 years now, but this is a nice review paper that joins all the papers that have been around this proposal, is that most areas of the brain are capable of keeping information in short-term memory. And the idea is that if this area has some role in processing the original information for perception or for action, these areas are also suited for keeping information in short-term memory. So this has come from evidence from functional MRI experiments, lesion studies, from electrophysiology in primates and in humans, and also from transcranial magnetic stimulation studies. So just as an example, since we are dealing with motion, this region here in the brain, they call it human MT+, but it's also called human V5+, which is the same area I showed you in the previous study. This area processes visual information of motion. When you try to memorize or keep information in memory about the direction of the stimulus or the speed, using fMRI, we are able to decode which direction is kept in short-term memory when we use this signal from this area to decode information. And we can do this with other areas of the brain for this specific stimulus. So usually those studies are done in working memories, investigating working memory, which is a bit different with the kind of task we have here. Working memory is when you are asked to memorize an information, such as when I ask you to remember my phone number and then call me so I can find my phone in the room. So this is the kind of memory that you attentionally and explicitly have to hold in your mind. But in our task, we are not asking participants to remember the speed of the target from the previous trial. We are just asking them hit the target as it hits the wall at the same time. So they are not keeping it explicitly. So they are implicitly storing this somehow. And we are interested to know which brain regions store this implicit information in the task we have at hand, in a visuomotor task. So we don't have just a visual task where the motion would be stored potentially in V5. But we have also other regions of the brain that are participating in the processing of the stimulus. So we are interested to know whether those other regions also participate in this storage of information. And we would like also to know if this previous trial effect rely on this trace of neuronal activity. It actually relies on it. So our hypotheses are that human V5, which we found on our previous experiment, and the dorsal premotor cortex, here written PMD, store information about the previous trial. So participants, just remember, they have to press the button when the target hits the wall. So they have to process speed, estimate the time, and press the button in the exact moment. So there is this possibility that the memory that is influencing the previous trial is about the speed, and there's also the possibility that it is the motor action that is influencing their behavior. And to test this hypothesis, we apply TMS, transcranial magnetic stimulation, which is a technique that I'm going to briefly explain later, but the idea is that applying TMS over those regions would decrease the effect of the previous trial if they are associated and affect this integration between previous information and current information. So we use TMS in a virtual lesion paradigm. It's kind of scary, but it is all according to the safety guidelines. So we're not doing anything that would cause harm to people. So the idea of TMS is that these here on the screen are neurons. They are all connected in this case. And the color that they have here is their current firing rate or membrane potential. So the normal processing of the brain requires a very complex interaction between those neurons, and they will have a state that will allow participants to process the information correctly. But when we apply TMS to participants, we induce electric field close to the brain, actually in the coil, which is the TMS. So the current will generate a magnetic field that will induce electric field in the neurons. And once we induce this magnetic field, electric field in the neurons, they will start to fire. They will start to fire synchronously. And this synchronous firing is already messing up with the normal processing of the brain. So what we are doing is we are not frying the brain, we are just activating a couple of neurons that might be participating in some process. And then by synchronizing them, we are disrupting the normal activation of the brain, which would be this complex interaction between the neurons. So here you see the colors are more close together, more similar, which means they are firing synchronously. And obviously we don't get all neurons, and we restrict our activation to a couple of centimeters, like 2 or 3 centimeters of cortex. We did this in 20 adults, young adults, and this experiment was performed at the Center of Integrative Neuroscience and Max Planck Institute of Biological Cybernetics in Tübingen. So the previous experiment was done here in Institute of Radiology at USP. So the task is very similar to the one I just showed you. So participants, they see a dot traveling from, would you like to ask a question, Galvis? Yes.
You used 20 volunteers.
Yes.
And how many times each one of them was submit TMS?
How many times they receive TMS on their head?
Yes.
So they, I have three stimulation conditions. So I stimulate V5, left PMD, and Vertex. They receive, for each one of those sessions, 255 trains of pulses, which leads to 65 trains of pulses in one session. So the task is very similar to the first one I showed you, just a couple of design differences here. Instead of 2 bars, I have like a box from which the target comes from. Participants, they have to fixate their eyes throughout the experiment at this cross in the center, and then the target comes from left to right, and the participant has to press the button at the same time as the target hits the wall. So here, the animation didn't work, but the target travels. You can actually see the target coming from the left to the right, and you press the button at the same time as it hits the wall. And in our experimental design, in order to mess with the activation of this putative trace of memory in the areas we stimulated, we apply TMS after the participants have performed the task. So we waited for 500 milliseconds so participants could press the button at the right time or close to the right time, and then we apply the pulse of rTMS. We repeated 5 pulses in 500 milliseconds, and then participants waited for more 3,500 milliseconds more. And this long interval is to make sure the effect of TMS that we are applying between trials does not interfere with the processing of each trial. So to make sure that the pulse of TMS would not mess with the processing of visual information or with planning of motor action, we had to make sure that the TMS was far away from the stimulus, according to previous studies, of course. So to find the regions we wanted to stimulate, we performed 2 functional MRI experiments. So participants were in the lab a couple of times, also to perform functional MRI experiments using the same images, the both signal that is a proxy for neuroactivation to find those regions. And to find them, we used 2 tasks. One very known task, which is stimulating the visual cortex with moving dots. So participants saw many dots moving in their visual field, specifically on their left visual hemifield, so we can find the right area V5, and contrasted this with static motion in the same region. So this helped us to find on each subject this area that processes visual information of motion. And to find the left premotor, dorsal premotor cortex, we used the same task as we used in the first experiment. So we gathered this knowledge from the first experiment to find the region we wanted to stimulate on this second experiment. And as a control region, we stimulated the postcentral gyrus of the participants, in this case the vertex, which is around here. So premotor cortex and human V5. Oh, we just stimulated the right human V5 and just the left premotor cortex. The right V5 because with the setup we had, V5 is able to capture information from both hemifields, so it wouldn't matter, at least this was our proposal, it wouldn't matter whether you stimulate left or right, and so we stimulated just one, and left premotor cortex because this would be the area responsible for planning the actions of the right hand. To analyze our behavioral data, so we collected data from temporal error. So 0 is the perfect behavior. So they have to press the button at the same time. So 0 is perfect behavior. Positive errors means that they are late. Negative errors means that they are too early, pressing the button too early. And we analyze these temporal errors with a multiple linear regression, where we have as regressors the speed of the current trial and the speed of the previous trial. And what we were most interested in was the parameter values for the previous speed slope, because if participants are biased by the previous trial, we would find in this case, in this situation, replicate previous studies, we would find a negative slope. So the greater the speed, the greater the anticipation of the participant. So this is what we would expect, a slope that is negative and different than 0. And in the case of TMS, if we are messing with this previous trial effect, what we would expect is the slope that is negative to start to be less negative and come towards 0. This is what we hypothesized and expected. So here I have the results. Here in the left, this is what we predicted from the beginning. So the idea that if previous trial has some effect on current trial, the faster the speed, the greater the anticipation on the next trial, and the same and the reverse for the slower speed. So here in the x-axis we have current speed of the target, and here on the in the y-axis and the x-axis we have previous speed of the target. So if we get just for the current speed 24 degrees per second, and we go further to the right, we see that as the previous, this is our prediction actually, as the previous trial increases in speed, our error comes to negative. So red values are negative, blue are positive. And the same thing here. If we go from right to left for the fastest speed of the current trial, as we go to the left, as this previous speed is slow and slow, we tend to be more late.
I'm not understanding. So you had 20 persons. This is what is an average is for every one of them.
Yeah, this one that I'm showing is our prediction. So it's neither mean or neither the behavior of one participant. This is what we predicted. This is our hypothesis. And on the right, but did you get what information I'm talking about here? I can explain in more detail this figure here. But this is our prediction.
I just not understand what is this. So you showed us in the previous slide a regression formula.
Yes.
Okay. So this is for one experiment.
This would be the prediction of the average response.
Show again the previous, please.
Yes, sure.
So let me tell you, there is a tradition here to ask a question.
Yeah,
today,
no
for some reason, nobody is asking any question, probably because we are transmitting. So, but we have 9 colleagues watching this. Please do something to help them understanding. So this. You got this taking data from one person?
Yes, I do a multiple linear regression for each participant, and I'm going to show you the data from all these regressions. Yes, so, and also if you're listening to us on YouTube, you can also make questions on the chat, right? So feel free to send questions. So this one in the left it would be the average temporal error of a participant, but this one is our prediction. Here on the right is the average behavior of all participants, and this here is the data from temporal error. So negative values are negative temporal errors, so people are going to be responding early, and positive errors are they are responding late. Microphone to Artur.
In this experiment of the no TMS, do you allow the 4-second interval between the 2 trials, just like you did with the TMS as well?
Yeah,
just to be sure.
It's the same inter-trial interval.
Okay, thank you.
So this is our prediction, and what we get here is very close resemblance from our prediction to the no TMS. So here we are just replicating previous studies, and we're just increasing the level of detail of what kind of effect we have here. So we have this gradual effect from previous trial as we compare every possible pair of trials preceded by 20, 22, 24, and 26 and 28 degrees per second. And one important thing is that, and I forgot to mention, is that every combination of previous and current trial have the same chance of occurring. So I have 25 possible pair of trials if I combine previous and current trial, and all of them have the same chance of occurring. So they are uniformly distributed. Now, when I apply TMS over the premotor cortex and compare it to Vertex, and Vertex is usually a control condition for TMS. But even if I compare Vertex to no TMS, you see there is a slight change of this gradual change in color. So it still goes from a more strong blue to a lighter blue here, and also from white to a lighter red here. So this previous trial effect seems to be present, but when we apply TMS during the inter-trial interval on premotor cortex, this pattern is somehow diminished. So we decrease this bias. This nice pattern of reducing is not that nice anymore. So for some of them, they actually look like they are stable. So this we call for somehow evidence that the TMS is messing with the activity during the inter-trial interval, which should be responsible for creating this bias. But even if we find this, here Here are the results from the beta, the slopes for the regression, Antonio. So here are the results for the regression. So each one of these dots are the slopes for the previous try regressor for each participant on each one of the conditions. So for no TMS, Vertex, human V5 plus, and PMD. Each dot is a slope, and here I have the average and standard deviation for these slopes. And what we see here is the difference between PMD and Vertex, and obviously between PMD and no TMS. This was also different. But we don't actually know what kind of information is being stored. So this is still an open question. Although we can interfere with the bias that occurs from previous trial, we don't know what kind of information is being stored in premotor cortex. The first obvious answer to this would be that there is motor bias being stored in PMD, and then we are dealing with motor memory. But that there is also two other possibilities. One, that PMD is storing information about time, because our task deals with time. We have to predict when the object is going to hit the wall. And participants, the brain might be storing this information. And this area has been previously implicated with time perception. And also with the perception of motion. So in experiments where you have to predict whether an object is going to hit a wall or not, we also have activation from premotor cortex, sometimes the ventral premotor cortex, sometimes the dorsal premotor cortex. So from these data, we don't have, we cannot solve this problem. We don't know whether it's a motor time or perception information. So this is an open question. Now, when we apply TMS over human V5+, we partially modulate previous trial bias. So, yes, Antonio?
By Claudia Vargas.
Sure.
So the questions are the following. So concerning the graphs you presented.
This one here?
Yes, I guess this one. Claudia make a remarks. She does not see so clearly the difference between both, and she asks you what is the statistics you are doing, and then there is a question: why didn't they use a real grasping action?
Real grasping action.
Grasping action. I don't know what she means by this.
I think she misspelled the S. I think they're just it must be grasping, right?
Okay, maybe.
Because it's easier to control the experiment using the computer. If we would use a grasping task, we would have to have cameras all over the lab and track the response of the participant with a hand. So, because it's easier to measure. There are some labs doing this, of course, but I think we opted for a simpler solution. Now, regarding the question about statistics, the statistics is here. The difference between both conditions, and we compare this, is the slope of the previous speed of the target, Claudia. So here is the difference between the conditions here. Yes, Ludmila.
I have a question related to Claudia's. So I'd like to know if there is any visual feedback of whether a successful grasp was made, because I would expect that the response, especially would be different from V1 to V5 with respect to that, because you would, with let's say, a closed loop experiment where you would have a visual feedback where your grasp is successful, you would have a different response or a different overall interest of the neural system to store the information if the grasp was successful or not, with respect to an experiment with no feedback, where the grasp, your attempt to grasp is a mere, let's say, exercise where the ball would like go straight to the wall or not, independent on your attempt. So I would like to know if you made any distinction between closed loop or open loop paradigm, and how would you expect it to affect your data on HV5 and PMD?
So regarding feedback, we chose not to show feedback on each trial to participants. So they wouldn't try to use these feedback, this explicit feedback, because they have feedback, and I'll talk about this in a minute. But we don't show explicit feedback about their timing. So they wouldn't try to use this explicit feedback to correct their movement, and just their own feedback. So although they are not actually catching the ball, they have an estimate. They should have an estimate of whether they were doing well or not on the task. Because we perceive the stimulus, we know when we press the button. Although it wouldn't be as precise as we would show them, you missed the target by minus 13 milliseconds. Now regarding the activity of PMD or HV5, that's another concern of ours. If we introduce another variable, like explicit feedback, we would somehow change how these areas might respond. And I agree with you, we would have to do another experiment to know the effects of feedback on this learning process and whether the explicit feedback would change the bias from the previous trial. So I would guess it would change the way they correct their movements. So they would be more precise. So I was about to present this one, right? I think so. Yeah. So when we applied TMS over human V5+, we partially modulate the previous trial bias. And by partial, I mean you can still see this nice gradient coming from positive to negative here in all current speed of the target. But it seems very close to Vertex, as Claudia mentioned. And when we do the statistics on the slopes of the regression, we don't find evidence for the difference between Vertex and human V5. They are very similar. Now, why is that the case? Is that a case that V5 is not storing information about speed in this kind of task? We are arguing this is not the only story that can be told here, because when we apply TMS over Vertex, we see a huge difference between not applying TMS and Vertex. And we explicitly moved the usual place Vertex is stimulated with respect to previous studies. Yes?
Question. You see, we see a huge. Why do you mean a huge? It's not that huge for us.
Yeah, okay.
So this is visually huge because you use a scale.
Mm-hmm. Yeah. It's huge compared to the other differences. But it's not huge, I agree.
No, it's not huge because it looks huge. It should be huge because of the probability of occurring is small.
Okay.
What is the probability of occurring this difference in this case?
Yeah, I don't remember.
So you see, I guess one of the problems that pictures does not tell you the truth.
Okay. So I'll remove the huge and there is a difference. There is evidence for the difference.
An apparent difference.
Apparent difference.
Because so the only way to state it in a rigorous way is to say, if I use this model, the probability of having this, if the model didn't change, is small.
Mm-hmm. Okay. So in this case, we didn't position the coil at the usual vertex position, which would be close to the supplementary motor area. So we moved the coil a little bit back to the post-central gyrus, which is associated with processing somatosensory information. And why did we do this? Because we didn't want to actually stimulate the supplementary motor area, which is also involved in motor planning. But it might be the case that the post-central gyrus is also associated with our task. And in this paper by Futre and 1 from 2005, they have shown that in order to estimate the position of an object and judge collisions between objects, the post-central gyrus is also associated with this kind of task. So it might be the case that we try to avoid and use Vertex as our control condition, but our control condition also messed up with the information stored in memory. It could be, it's a long shot, but it could be the case. And when we compare V5 with no TMS, we also see a difference here. So V5 might still be storing information in short-term memory. We just don't see evidence here for the difference between HV5 and Vertex. And why would this be the case? One possibility is that information is stored in both human V5 areas. So as I said in the beginning, our stimulus set was showing, just finish this thought. Human V5 was able to process information from both hemifields, so we would see the target from both hemispheres. So it could be the case that we are stimulating the right V5, but while we are stimulating right V5, the left V5 is also keeping information from the previous trial. So the bias is not completely decreased or removed because there is still information from the previous trial on the left side. And when we remove the TMS, these two areas are still talking, they are still wired, and one might transfer information from one to the other. So this is one possibility. And before I go to the next two, I will let you, or who else has a question?
No, it's just a question, just to be sure. It's Claudia wants to know if it is a sham TMS?
No, it's real TMS over Vertex. It's not sham. So sham, for those of you, sham is when you just tilt the coil on your head and you don't actually stimulate the head. It's just the clicks and noise of the TMS. And participants, they don't feel the sensation because it stimulates some thin muscles here and you sense the contraction. It's not really pleasant. And when you just tilt it, the effect of the TMS is completely over. You don't have the stimulation, just the noise. So no, Claudia, it's not sham TMS, it's real TMS over Vertex. So another possibility is that, well, V5 is not participating in the storage of information in this case, but there's a top-down modulation. So areas along the hierarchy of processing the stimulus keep the information about speed, and when the upcoming speed comes, these areas from along the hierarchy, like MST, feed back information to V5. So both informations can be fused, so current and previous speed. So this is also an open possibility. We don't actually know from our data if this is the case. And the other possibility is that the information is stored in a hidden state. So there is this nice paper by Zokai where she manipulated the working memory of participants, some things that were presented, two items presented at the same time. But after these two items were presented, she asked participants just to remember one of them at one part of the trial. And then after a couple of milliseconds and after performing the memory task on the remember target, she asked participants to remember the other one and apply TMS between those trials. And what she found was that when applying TMS to mess with the information of the not attended target, she can't find evidence of perturbation. So it might be the case that the information is stored in a way that is protected from the activation of TMS. So one possibility is that the balance of the neurotransmitters, of ion channels, the ions around the synaptic button make sure that interference from outside or from TMS or from other areas keep information from this interference, protect the information from this interference. But also this is also speculative, and we are trying just to explain what are the possibilities for these results. So our conclusions from these experiments, is that previous trial effect relies on an implicit short-term memory mechanism, stronger evidence for PMD, but still not so clear for human V5. And that the regions within the processing hierarchy of the stimulus, they have a role in keeping information in implicit short-term memory, and not just working memory or explicit memory as has been shown before. So these are main conclusions of this experiment. But still, of course, we have many things we want to learn about this potential mechanism. And one question we have is how the representation of short-term memory affect the processing of information on the current trial. We've just shown here that it somehow does, but we don't show it how. And we are also not sure how TMS decreases previous trial effect. Although TMS increases the noise of the processing of the information, we don't know exactly how this noise introduced in the system causes the next trial to not be that affected by the previous trial. So one way to think of this problem is using probabilistic coding. So this is one idea, that the brain encodes probability distributions. So in our experiment, this would mean that each speed that participant experiences would be stored in the brain somehow as a probability distribution, centered with mean around the actual speed or with some variability around this encoding, and also with a variance on the stimulus representation. So the idea is that the distribution with which the stimulus is represented, here using a normal distribution just because it's easier, the stimulus would be encoded in this using distributions, probability distributions. And then the precision of this encoding would be the inverse of the variance of the stimulus. So if I say the precision increases, I'm saying just that the variance is decreasing. And if we talk about memory, it could be the case that if we store, as we perceive the stimulus, we store it as a probability distribution. As time passes, the precision of the memory would decay. Not the mean, but the variance would decay. So the idea is that the current information would have higher precision, and the memory information would decay here with an exponential function, but it would decay and increase its variance or decrease precision. So this is an idea. And one nice way to try to integrate both information. So information would be first encoded as a probability distribution, and then it would decay with time by decreasing its precision. But then when the next information comes, the system would try to estimate the current speed by doing a weighted average, in this case using Bayes' rule of the current and previous information. And this weighted average depends on the precision of the target, or depends on its variance. So the greater the variance of the memory trace, so the longer time it decays, more the information would be close to the actually sensed stimulus. So in order to combine those information, we can use Bayes' rule to get a new estimate of the actual, of the sensed stimulus. So this would be the idea of how this integration of memory and current stimulus would occur. Pergunta aí, oh, no, sorry.
I'm trying to follow, but the question was a question, but it's too late now.
Okay, there's a 20 seconds delay, so yeah.
And my reaction is a little bit longer. So.
Okay, no worries. So if we use this Bayesian Ideal Observer models, where we get probability coding by each stimulus and also combine this information using Bayes' rule, how TMS decreases serial dependence? What is the case here? The idea I'm trying... Yes, Aline?
Sorry, can you come back? What is the difference between the estimate and your current information?
The idea is that the current estimate is what the sensory system has, from retina to early visual cortex.
I'm sorry, from a mathematical point of view.
Oh, okay. The difference between current information and estimate is the current information is what leads to the estimate. So in order to get the estimate, you need memory and current information. You need both, unless you don't have any memory.
So your estimate is your speed, or not?
Yeah, the estimate
is in time k.
Yes.
Or not? Oh,
yes. Okay. So for speed in time k, which would be the estimate, yeah, the notation is wrong here. So I didn't use k and k-1 before. I used to use speed estimate and speed current. So now it's confusing. Yeah, it shouldn't be k here, because here is the current information and here is the estimate. Yeah, sorry about that.
Okay.
So here is current information and here is the estimate. Yeah, I forgot to change this one as well. Okay, so to get the estimate, you need both memory and current information.
And your speed k-1 is your memory?
Yes.
And your current, okay.
Speed k-1 would be the memory, the mean of the memory, and the standard deviation, the variance is here. So how TMS decreases serial dependence? The idea, as I can show change in the slides, is that it would decrease the precision of the current memory trace. So from one trial to the next, if I apply TMS, the curve would be more broadened if I didn't, compared to when I don't apply TMS. And then the estimate gets closer to the actual stimulus. So that, yeah.
This is what you're expecting to happen, or
Yeah.
this is in
This is
your data?
This is what I expect to
okay.
be happening using this model. So these would be the predictions of the model, although I have already seen my own data, and that's actually what happens, right? From the results from the slopes. So yes, this would be a possible way to explain how TMS is affecting. It's changing the shape, the scale of the one of the parameters of the distribution, the probability distribution that encodes the memory. So once again, I have here, I've run some simulations and predictions, and I'll show you it compared to my data. So on the left, it's different from that previous one. The other prediction was not a simulation. I just painted the squares the way I wanted. Here is the simulation using those equations, and how, by presenting to these equations or for this simulation the same stimulus sequence as the participant has experienced, how would these equations behave? And they behave very similar to the no TMS condition. So with a decaying rate of 0.1, what I get is a very close response to no TMS. So as I get further to the right, participants tend to respond earlier, or a Bayesian Ideal Observer tends to respond earlier. And each speed has an influence on the next one. No? No. I saw a movement on my periphery already on Startle. So also to evaluate the simulations, I measure the behavior of the ideal observer with the same metric I use to evaluate my participants. So I estimated the slope of this multiple regression for the previous speed as the decay rate increases. So as the decay rate increases, the slope would decrease, which is what we observe, and I already show you this graphic. So the idea is that by applying TMS, we are increasing the decay rate of the exponential function that deals with the memory trace. Here I'm not telling that I could tell that the time is going faster, the decay rate is not, but the participants have the same time to perform the task. So time is fixed for all participants. So I don't know if I could test the hypothesis that the time of decay is actually passing faster, or if the decay rate is increasing. I should probably do some model fitting and see which one has a better explanation of the data, but I haven't done this yet. Now this model makes a very interesting but weird prediction for the slope of the current speed. So the prediction is, as the decay rate of the memory trace increases, the weight or the association of the current speed and the temporal error decreases. So what would you expect if I tell you that the memory trace is being vanished and I do this regression? Wouldn't you expect that the participants are relying more on the current speed information and actually increasing the slope of the current speed? So I'm removing information from the past. Shouldn't be the case that participants are relying more on current information? So this model predicts otherwise. It predicts that as the decay rate increases, in other words, as the memory trace diminishes, there's a lower association for this analysis between decay and current slope. And this is actually what happens here on our data. So when we look at PMD and V5, there's a decrease in the slope of the multiple regression for the current speed regressor. So this is somehow weird but interesting. It might be the case that when we remove the bias from the previous trial, on every trial, participants are not biased on average. And this average bias that would be causing participants to be way faster on the slow speed and way later on the fast speed.
Sorry, because maybe I missed something, but what do you mean by current slope?
Sorry, that was my. I think the words are not good. It's the current the slope of the current speed regressor. So the regression has two regressors: the speed of the current trial and the speed of the previous trial.
Yeah, and
the slope here would be. I would expect it to be positive. So as speed increases, participants tend to be later. So the faster the target goes, participants tend to go to be late with respect to the target. They respond later. Mm-hmm. So this would be a positive slope. And what happened here is that the slope, or the association between the current speed and the temporal error, decreases as I apply TMS. So it goes closer to zero. Not exactly at zero. You see it starts to asymptote here around 0.7 for lambda, but it doesn't asymptote at zero.
So what you have this current slope is beta 1.
Yeah, beta
1 hat.
Sorry, yes. Current slope is beta 1. Yeah, that was my mistake, yeah. Sorry. Is that clear now? Okay. Sorry. So this is what happened. We're still curious to know why this is the case, but one possibility is that on average they are not biased by the previous trial, and then although they rely on current information, that would be very weird if they were just closing their eyes and knowing the speed of the target. But the bias that participants experience on average is also caused by the memory trace. And then when they face a very fast target, when we remove the trace, they are not as biased on average. And then that's because the current slope would decrease. But that's still, for me, it's still very weird prediction. And actually, the fact that prediction is met here is also very weird.
Maybe I missed something. As she said, what is the difference between current slope and the slope current?
That's also a typo mistake of mine. It's also current slope. Sorry.
But I still don't get it, because you have exponential in your second term, no?
Here?
Yes.
No, this one is this is not part of the model that is simulating the data. This is the analysis I'm using to evaluate the model behavior, how the model behaves after experiencing the trials.
So what do you mean by decay rate? So because I understood that your decay rate would be the rate of your exponential, how fast you are losing memory with respect to your beta 2. So now I don't get your model.
Yeah. So maybe I should come back here. So this is the model that I'm simulating to respond to the targets that I present to participant. In this case, the computer. The decay rate that I'm changing here is the decay of the stimulus the distribution that is encoded in the trial. So I encode the trial and I apply the decay rate to get here.
Your memory, which is encoded in your speed in the previous step.
Yes.
So it's like having an exponential in your second term, no?
But not for beta 2, because beta 2 is not part of the model. That equation is what I'm using to evaluate the response of the model.
Yeah, but you simulate your model following the previous formula, no?
Yes, but this equation here is not part of the model.
Yeah, I know, but you use that formula to simulate your model. This is why I'm not understanding how can you find this decay rate.
This one here.
Yeah.
So these are 7 simulations. I think here I have 7 different simulations. When I simulate with decay rate of 0.1, this is the kind of slope I get.
and your decay rate is that exponential in the
Is that
previous
exponential?
formula.
Yes.
Okay, this is why I'm not understanding, because you're saying that as your decay increases, you are putting much more weight in your beta 1. Less?
Yes.
Wait, this is...
I'm putting...
I can understand. How can you find this? This is my question.
Yeah, me too.
Okay.
This is the counterintuitive thing about this prediction. The prediction of the model is that the beta 1 is going to decay even though it should be increasing. That was my first impression. Okay, if I'm taking information from memory or messing with it, I should increase beta 1 and not decrease it. But it decreases. It decreases, and my, I think, sketchy explanation is that removing the memory trace from every trial makes them less biased on average. And this is what might be causing they are less reliant on the current information. They rely on the current information, but since I'm taking the bias from the previous trial, they don't have so much high errors for instance, the too fast speed or too slow speed.
I have a question by Claudia.
Sorry, guys. Just to make sure that I explained it correctly. So I have here like 7 different simulations. The decay rate of the model would predict is a prediction of how these would behave on every simulation, but I don't have a decay rate here, which I think was what you were asking.
My question is, if you are if you are increasing your rate of loss of memory, you should have much more weight in your current speed.
Yes, I agree with you, and
but it's a simulation. So in simulations, usually we can find what we believe it. So
yeah, but this is what the simulation predicts, and
We have several questions.
Okay. We can discuss it later, Aline, or see if those questions are also related to this one. But I think I have to make it more clearer.
We have a question from Marco Dumas Gubi. It's from our team, and he wants to know if removing the bias would be like if the patient would start afresh. Or this is a statement, not a question. But the question is how this is different from starting afresh.
From the data we have, they don't actually start fresh. We decrease the bias, but they are all still a slope that is different than 0. So the bias is still there, we just decrease it. So they are not starting fresh, we're not refreshing their memories. So I think this is a difference.
So there is a question by Claudia Vargas.
Okay.
So that's a question she asked before, and then, But then she said, I will translate immediately. You need a control on TMS applied to a certain... You need to check that the TMS applied to a certain region does not interfere with the task is going on. Otherwise, this does not correct. You don't have evidence that you have a specific interference on the short-term memory. So the question is there any brain region for which there is no effect of TMS in this paradigm? So I say it again, is there any brain region for which there is no effect of TMS in this paradigm?
I would guess there is. I would just have to find a good area probably, because my take was that I should stimulate Vertex, because Vertex is usually this control area that shouldn't have activation. That was a misplanning of ours. We thought Vertex would be our safe control stimulation region, but it wasn't. We have no TMS still, but yeah, this is a really complex task if we think of brain processing. So I would have to find which brain region would not be associated with it first, doing many experiments. So yes, this is a limitation of this study. We thought our control condition would be the best one. It wasn't. No more? You said you have several questions. Oh, okay. You see, several is... So this is still work in progress. So since I finished my PhD, I've been trying to learn computational cognitive modeling, which is something new to me. So I so far have done a couple of simulations with different variations of the model. And I'm still trying to fit the data to the model, which is, I think, something very important. Not just to simulate how the what is the behavior of the model, but fitting the data to the model to our data. So this is something ongoing and which I'm learning. And other things is that while I'm doing those simulations, I'm making more predictions of what kind of variations of the experiment we can do to make sure this model is actually explaining our data. So we are designing currently new experiments to test these new predictions. So two of those predictions are, one of them is very intuitive, that longer inter-trial intervals, so as we space the time between trials, the bias from the previous trial should decrease. So instead of messing with the decay rate by applying TMS, by doing a simple behavioral experiment, we should, by making sure participants have a long time between trials, making it very, very boring, more than it already is, we should decrease this bias. And we are already collecting data at the Federal University of ABC, which is really nice to say it in English, in collaboration with André Cravo, which is a professor there, and Cristiano Azarias, which is a great undergraduate student. So we are now a couple of participants so far, but we are collecting this data at the moment. And another prediction that this model makes is that the uncertainty at the encoding should modulate the bias on the next trial. So if this probabilistic coding idea has some resemblance with what the brain does, we should see that more uncertain stimulus should lead to less bias on the next trial. And what do I mean by more uncertain stimulus? There are some kind of stimulus that you can present to the brain that you are uncertain about the nature of it. So if I present to you my two fingers on a foggy environment, you are uncertain about how many fingers I'm holding. If I do it a little bit twisted, you cannot see my two fingers. You can actually see just one of them. So these are uncertain inputs to our system. So I can manipulate the uncertainty about the speed of the target by changing its contrast. It has been shown that if you decrease the contrast of the stimulus, you tend to be more variable in your responses and actually respond as if the target is going slower. So I can manipulate the uncertainty of the stimulus, and by doing so, this would lead to the prediction that targets that have higher uncertainty would lead to less biased behavior. So this is still in planning stage, and I will probably do it here if time permits. And yes, question.
Yeah, it's still about the fresh starting. It's Gubi wants to know if it's possible that this remaining bias somehow has an enhanced contribution than expected.
A what? Sorry. I didn't understand the final part of the question.
Enhanced contribution than expected.
Unanswered? I don't get it.
How you read it?
Unexpected contribution? I don't know.
Enhanced.
Increased. So something which is stimulated,
Enhance the behavior. Okay. Thinking about the model, if it was increasing or enhancing behavior, we would expect to have less variance on the information. So memory trace would be improved and not decreased. And we don't see it on the data or in the predictions of the model. So I know there are some TMS protocols that are designed to improve behavior or increase excitability of the cortex. But in this case, we are introducing, what we expect to be doing is to introduce noise in the system. And this noise would be detrimental to the behavior and not increase behavior. Challenges, accepted or not. We still don't know how short-term memory is stored in the brain. This is still a great challenge, and there is a lot of debate about this. There are people thinking that higher order brain regions like the dorsal prefrontal cortex or interparietal sulcus store information in memory by using continuous spikes that would store this information, and that there's a lot of other researchers thinking about that in order to store information in memory, you don't need neurons activated all the time. They can be taking turns with other neurons in the system, and there's also the possibility that information in particular in short-term memory, not in long-term memory, could be stored by short-term synaptic plasticity, which would be something like changing membrane currents or concentration of calcium inside or outside the synaptic button, or the concentration of neurotransmitters. So this is already a very heated and hot topic on the field.
Concerning the second point, which is very interesting, to the best of my knowledge, we don't know absolutely how the brain does statistical inference of whatever. At most, we succeed getting evidence that something which is similar to statistical model selection happens. But how? To the best of my knowledge, nobody ever succeeded. Not even for simpler things like present the same tune 5 times and then change last time and then you observe. But what happens? Is that true?
Yeah, to the best of my knowledge, I know there is people seeking this to answer this question. And so far, I don't think there is a very concrete answer. But I think there is a lot of proposals about how it's done. So some people think that it might be encoded in the firing rate of one neuron or in the population of neurons, or that it's not the probability distribution, but the log of the probability distribution that is stored in the brain to make sure that inference is made easier, because if we have the log, it's easier to make multiplications. If you have probabilistic distributions, it's easy to sum but not to multiply. So I think 2 challenges here. One is whether population probability coding exists, and the other challenge is how inference is done. So in order to perform inference, you need to code probability. So these 2 are real challenges to the field, but I think there are groups that are working on those challenges. I think mainly you guys, of course, we guys, I have to include myself now, I shouldn't forget this. But I think one lab that, 2 labs actually I can remember from the top of my head. Alexander Pouget in Zurich and Weijie Ma in, I think, New York University. They are working on this problem. I should probably read more about their papers, but this is something I haven't explored that much. So I would really like to understand more about this problem as well.
It's an open question since von Helmholtz asked about this. So that was 140 years ago. So it's a difficult question.
And to make things more difficult, both problems at once should be also a nightmare. Because if we are trying to explain short-term memory as probabilistic coding and as inference from probability distributions, it should be even harder. So these are challenges that are really, really interesting, but really hard to figure out from where to begin. So Aline, I would like to ask you how much time I still have. It's all day? No, it's over? Okay, I had a couple of more slides to present, but I can present it other day. It's no problem, no big deal. So, okay. Okay, so thank you all for your attention, and I'll skip for the acknowledgements. You didn't see anything before. I'd like to thank both my PhD supervisors, so Edson and Andreas, André and Cristiano for their collaboration on the ongoing projects, Antonio Galvis for the opportunity here to be part of NeuroMat and start. The other part of the talk was about NeuroMat, but okay. And thanks to CAPIS for the support and FAPESP.
Do we still have questions?
I'd like to ask a question about the first result slide.
Okay.
And it's with respect to the asymmetry that apparent asymmetry that we observe. Can you show us your expected matrix and the obtained matrix with no TMS?
Ah, for the second experiment. I thought it was for the first one.
Yeah.
This one?
Yeah, so in this one, there's like, okay, an apparent slight asymmetry between vertically, right? I'm sorry, horizontally. So the current speed, the higher the current speed is, and the lower current speed is, the worse the prediction, right? It's like so the predicted matrix is perfectly symmetrical, but with no TMS is not symmetrical. Like your intake on this, and I would have a guess that you could, for the subject could, for instance, be taking into account the expected value, absolutely expected value of speed. So we would have a little bit of weight on the, I don't know, 22, 24, around 22, 24. So it wouldn't really mess if you guess a little bit on the expected value, something like that.
So this asymmetry that happens here is a very interesting phenomenon called central tendency, which speaks directly to what you're saying. Participants expect stimuli to be around the average of the distribution. So in this case, the average would be 24 degrees per second. And the asymmetry here, it happens because of this. So as I go further to 28 degrees per second, even if I present 2 times in a row, 28 degrees per second, as they are expecting somehow, they might have learned the distribution of stimuli. They do not behave as if the target was repeated. So there is a bias from the previous trial, but there is also another bias that I haven't talked about, which is the central tendency. And the model I showed you, I haven't showed you this part of the model, but I am also modeling central tendency, which is another very interesting phenomenon here. So participants tend to rely on 2 information, not 2, 3, current information, previous trial information, which has a great bias, and another one is central tendency. When I simulate without central tendency, this is what happens. Now, when I simulate that participants are also relying on an average response, this is what happened. It's a little bit weak here, but you see it's not completely white, it's not 0. And here it's not completely 0, it's also a little bit reddish. So bluish here, reddish here. So I have to take into account that they might be relying on some distributional information about the set of stimuli, but also the bias. If I remove the bias from the previous trial, the model doesn't work as well. So both should be needed. No, it's not. Yeah.
Alô? Agora sim. Se eu não me engano, no momento você fez uma distinção. Sorry. If I'm not mistaken, at some point you made a distinction between short-term memory and working memory. But I got a little lost in the end because, from what I understand, the findings in the storage of memory encoding without using the spikes is the findings I know is for working memory. And your study was short-term. So you think it's also short-term memory being stored in the, well, like you said, calcium in the membrane?
Well, I don't know. So first to make sure this is clear. Short-term memory is one component of working memory. Working memory also relies on executive control And you have to manipulate information and explicit information, so also relies on attention. The way I'm studying short-term memory is without attention during the period I don't have any task. So this would be the difference. Now, for studies of working memory, there's still an ongoing debate whether information is stored in higher order areas in prefrontal or parietal cortex, or whether it's stored in sensory regions or motor regions or elsewhere in the brain, like the other slide I showed you. Now, if in these sensory regions, if it is stored as a spike train, as a dynamic spike train between all neurons in the network and not just a couple of them that always are spiking or membrane potential, these are all under debate. And the one about the membrane potential is still has just a few evidence from experiments, and most of the work is done by modeling. So there is one very nice study where they asked participants, like I said, to remember two things at once, and then they ask, okay, just forget about this one. And then they, using EEG, they try to decode what stimulus is being stored in short-term memory, in working memory in this case. When you apply TMS to the area that is potentially responsible for this storage, you can redecode the information that is not being attended. So even if you are encoding this, you cannot pick it up with EEG. But if you apply a pulse with TMS, it is as if there is information there. And then when you spike the neurons, they merge with the spiking that resembles the information that is stored in possibly membrane potential, calcium imbalance, neurotransmitter imbalance. I don't know exactly which one. I think it's a fruitful line of research. Many people are working on it.
I have a question. Because actually what you have in your data is 25 pairs, right? You're making an average between the 25 pairs.
Yeah, for these graphs here.
Yes.
And, yeah.
And how many trials do you have for each one of these pairs?
For each TMS condition, because I have no TMS, Vertex, PMD, for each one of them, I have,
in each graph.
Yeah, I have 10 trials per pair.
So
I have
each one of these
250.
colors is an average in 10.
Yeah.
Okay.
Yeah. Not for these no TMS, because it's the average of the averages. It's the average of all participants.
Okay. So you have 20 times 10, 200.
19. I have to eliminate one participant. Yeah.
Okay. And the other question
exclude,
is...
not eliminate.
Hmm?
I used the wrong word. I said eliminate. It's exclude participant.
And how do you present the stimuli? Do you have a fixed sequence in these speeds?
No. It's a different sequence for every participant.
And how do you choose this sequence?
I used an algorithm developed by another researcher that makes sure that every pair happens with the same probability as the others, but then you can shift them. But
there is no relation, they are not increasing or decreasing, right?
No, it's same chance of occurring each speed and having each one of those pairs preceding each other.
I saw this before. I'm looking at this now. The error is really different between your the simulation and your actual data.
Yeah, this is probably because
your variation.
I probably didn't set up the noise of the model to be according to my data. So since this is simulation, I didn't tweak the parameter to make sure it fits the data. So I guess if I fit the model to the data, I would get a closer variation for the model and for the participants, I guess. So here it's 70 milliseconds and minus 70. So it seems that my model is behaving with higher variability than my participants. Yes, but I guess if I fit the model to the data, it will behave closer to the participant, I guess. Okay, should we wrap up or any more questions? Okay, thank you very much for everyone who listened.
Thanks again.